In 1914, John S. Dexter noticed the appearance of a notch in the wings of the fruit fly Drosophila melanogaster. In 1917, Thomas Morgan Hunt identified the notch alleles. Notch signaling is essential for embryonic development and tissue homeostasis. Because both notch and notch ligands are associated with the cell membrane, notch signaling occurs via direct cell-to-cell contact. Notch is an essential regulator of cell fate. In this example, the division of a cell, for example a stem cell, results in two daughter cells, which differentiate into separate lineages. In the parent cell, a negative regulator, shown here in yellow, is asymmetrically distributed. Upon division of the parent cell, the negative regulator, for example the protein NUM, inhibits notch signaling in one of the daughter cells. Thus, notch signaling is activated in only one of the two daughter cells. In this way, the different fates of the two daughter cells are determined. Notch can also amplify slight differences between adjacent cells. In the present example, the intensity of the blue color indicates high notch activity. Initially, notch signaling is active in the center cell as well as in the peripheral cells, with notch signaling activity only slightly higher in the center cell. Later, signaling becomes unidirectional from the center cell to the peripheral cell, leading to varying cell fate decisions. A similar pattern of notch signaling is observed in the proneural cluster of Drosophila. Notch signaling can define boundaries between different cell populations, for example, in the fly wing primordium, as shown here in the example. The mammalian genome encodes 4 highly homologous notch proteins numbered 1 through 4. Here we see notch 1. The notch signaling pathway is characterized by a sequence of proteolytic cleavage steps. In the Golgi apparatus, the mammalian notch precursor is already cleaved, resulting in 2 fragments held together by non-covalent linkages, the notch extracellular fragment, or NEC fragment, and the notch transmembrane and intracellular fragment, or NTMIC fragment. Notch proteins contain various domains, indicated here by different symbols and colors. The N-terminus of the extracellular fragment contains a number of EGF repeats. The EGF repeats involved in ligand binding are shown here in red. The negative regulatory region involves C-terminal amino acids of the extracellular fragment and N-terminal amino acids of the transmembrane and intracellular fragment. A crucial regulatory point in notch signaling is ligand-induced and atom metalloprotease-mediated notch receptor cleavage to release the extracellular fragment. The negative regulatory domain prevents proteolytic cleavage in the absence of ligand binding. Non-covalent bonds of the heterodimerization domains link the extracellular, the transmembrane, and the intracellular fragments to each other. The transmembrane domain contains cleavage sites for gamma secretase. The nuclear localization signals guide the intracellular notch fragment into the nucleus, and the ankyrin repeats mediate interactions with other proteins. The PEST domain mediates the degradation of the intracellular notch fragment by the proteasome. As mentioned before, proteolytic cleavage plays an important role in notch signaling. Here we see the different cleavage sites. At site S1, the notch precursor is cleaved during biosynthesis. S2 marks the site cleaved by atom metalloproteases, and S3 and S4 are the sites cleaved by gamma secretase. Mammals produce 2 groups of classic notch ligands, the jagged ligands 1 and 2, and the DLL ligands 1, 3, and 4. Here we see the domain organizations of jagged 1 and DLL1. The DSL domains as well as the DOS domains are involved in receptor binding. Jagged ligands contain a cysteine-rich motif, but DLL ligands do not. Ligands of both groups are anchored in the plasma membrane by their transmembrane domains. The Notch signaling pathway. Notch signaling is initiated by the binding of a Notch ligand to the Notch receptor present in adjacent cells. It is thought that ligand binding to Notch combined with the start of ligand endocytosis induces a conformational change in the negative regulatory region of Notch. The S2 cleavage site becomes accessible for proteases of the atom family, which cleave the heterodimerization domain of the Notch transmembrane and intracellular fragment. The complex of Notch ligand and the Notch extracellular fragment are endocytosed by the ligand-expressing cell. After shedding the Notch ectodomain from Notch, the transmembrane domain of the remaining Notch fragment is attacked by a gamma secretase, a member of the multi-component complex belonging to the family of intramembrane cleaving proteases. Cleavage starts at the S3 site. As a result of cleavage, various amino acids may be present at the N-terminus of the Notch intracellular fragment. Only the fragment with an N-terminal valine residue escapes N-end rule degradation by the proteasome. The Notch intracellular fragment is liberated. Cleavage then proceeds to site 4. As a result, a small N-beta peptide is released from the lipid bilayer. The Notch intracellular fragment, which has been released into the cytoplasm, enters the nucleus. In the absence of Notch, the CSL DNA-binding protein associates with co-repressor proteins. In the absence of Notch, transcription of Notch target genes is repressed. Notch itself does not bind to DNA. Instead, Notch associates with CSL. Upon Notch binding, the co-repressor proteins are released from CSL. The complex of CSL and Notch binds the coactivator mastermind, abbreviated MAM. After recruiting coactivators to the complex, the multiprotein complex induces the transcription of Notch target genes. The Notch1 gene is mutated in over half of all T-cell leukemias. The reciprocal translocation between chromosomes 7 and 9. The important role played by Notch1 in T-cell leukemia first became evident through the analysis of the reciprocal translocation between chromosomes 7 and 9, leading to the translocation of the Notch gene into the T-cell receptor beta locus. As a result of the translocation, a promoter of the T-cell receptor locus drives the transcription of the Notch1 gene. The 7-9 translocation is rarely found in T-cell leukemia. More often, mutations are present in the heterodimerization domains or in the PEST domain of Notch1. Mutations in the heterodimerization domains facilitate cleavage at the S2 site, independent of ligand binding. Subsequently, the transmembrane domain is cleaved by gamma secretase. This liberates the Notch intracellular fragment, inducing transcription of Notch target genes. Mutations in the PEST domain lead to its truncation. The Notch intracellular fragment can no longer be ubiquitinated and is thus no longer degraded by the proteasome.